Good afternoon, everyone. I welcome you all to the afternoon session of doctoral students. So today, chairing the session, we have with us Professor Rajagopal Raman. He's a professor at the Department of Zoology, University of Delhi, and his areas of interest are gut microbiology and vector biology. Dr. Raman has done his PhD from IARI New Delhi. He was a scientist at the Indian Council of Agricultural Research. He was a staff scientist at ICGEB New Delhi. He has received Scopus Young Scientist Award and he was selected for the MR4 International Bioinformatics Workshop on the Malaria Vectors 2003 at Johns Hopkins University. Our other chairperson is Dr. Srijit Parameshwar Panikar. He is assistant professor at the Department of Zoology, University of Kerala. His areas of interest are tropical cutaneous research, hair biology, cancer biology, Ayurveda biology, stem cell biology, and developmental biology, among many others. Dr. Srijit has done his PhD in 2007 from University of Kerala. He was the research associate at the Department of Dermatology, Case Western Reserve University, Ohio. He did his postdoctoral fellowship from Department of Cancer Biology, Cleveland, Ohio. He did his postdoctoral research associateship from Department of Cancer Biology from Chicago, Illinois. He has produced several documentary films on environmental related issues. He has produced a documentary series, Sastra Vidi, for All India Radio. He is member of Society for Investigative Dermatology, North American Hair Research Society, Society for Reproductive Biology and Comparative Endocrinology. I welcome you, sir. Starting with the session, I would like to hand over the mic to the chairs. I would request all the participants to please stick to the time limit, which is 8 minutes plus 2 minutes. Thank you.
Good afternoon. This is doctoral students award session. There has been a request from that suggests kind of like okay, I request to get the presentation first. I hope everybody else is agreed to it. So, Dinesh, we will be talking on targeting of human metabolism by trans This session has a maximum audience.
One second. कनेक्ट हो रहा
respected chairpersons, and my dear friends. I am Mithilesh Kumar Temre, doctoral student of Professor Sukh Mahendra Singh from School of Biotechnology, Banaras Hindu University. Today I am going to present my work entitled Targeting of Tumor Metabolism by pan-GLUT inhibitor GLUTOR. As we all know, this cancer is a devastating disease. It is a disease of concern affecting millions worldwide. Also, it is the second leading cause of death. Hence, there is a greater need for development of new anti-cancer therapeutic strategies. As neoplastic cells display altered carbohydrate metabolism, in which they show overexpression of glucose transporters and also increased glycolysis, producing lactate for their bioenergetic needs as compared to normal cells. Hence, targeting GLUT has been emerged as a new anti-cancer therapeutic strategies. In this study, we have used a newly identified pan-GLUT inhibitor, GLUTOR, which molecular mechanism has been not known so far. The experimental approach included in silico tools for molecular characterization of binding of GLUTOR to GLUTs, along with in vitro and in vivo investigation for characterizing the molecular mechanism of its antineoplastic action. This is the schematic strategy for the in silico analysis. The in silico study shows that GLUTOR strongly binds both the prominent active site of GLUT1 and GLUT3. Also, there are various non-covalent interactions involved during docking, which are hydrogen bonding, hydrophobic interaction, ionic interaction, and van der Waals forces. They cumulatively reinforce the binding of GLUTOR to both GLUT1 and GLUT3. Also, the binding of GLUTOR with GLUT1 was stronger as analyzed by using YASARA and patch docking server tool. The GLUT1 GLUTOR complex was further analyzed by molecular dynamic simulation and trajectory analysis, indicating various parameters. The dynamic simulation and trajectory analysis shows that the binding of GLUTOR to GLUT1 was thermodynamically stable throughout the study. I have also done trimeric docking, in which we used GLUTOR, glucose for docking with both GLUT1 and GLUT3, and found that involvement of various common interacting amino acid residues, which indicate GLUTOR could be a competitive inhibitor of glucose binding site. But further experimental approaches need for the prove the hypothesis. The key finding of the in silico study shows GLUTOR can stably bind to the active site of both GLUT1 and GLUT3, and also overlap with the binding site of glucose. Also, the GLUTOR-GLUT1 complex was thermodynamically stable as analyzed by simulation analysis. Therefore, results suggest that GLUTOR may be a competitive inhibitor of glucose and could act as a potent inhibitory action. Next, to prove the hypothesis and validate the in silico investigation, we further analyzed in vitro and in vivo investigation to check the anti-neoplastic action of GLUTOR, for which I have used a thymoma model designated as Dalton lymphoma as a tumor model. This is the experimental procedure for in vitro experiments. In the in vitro part of this study, the result shows that tumor cells display a higher expression of both GLUT1 and GLUT3 as compared to their normal counterpart thymocytes. After that, GLUTOR treatment to tumor cells show dose-dependent and time-dependent inhibition of cell survival, along with augmentation of the apoptotic cell death. Next, to validate the in vitro study, we have analyzed the in vivo investigation by administering GLUTOR to tumor-bearing mice, followed by analysis of various tumor progression, tumor load, and survival of the tumor-bearing host. The in vivo investigation shows that administration of GLUTOR to tumor-bearing mice retards tumor progression along with survival of the tumor-bearing host, in which male respond better compared to female, as analyzed by various parameters of tumor load and tumor progression. Next, in order to understand the tumoricidal action of glutar in vivo and in vitro condition, we check the expression of various key metabolism and cell survival regulatory molecules, in which we have checked the expression of, as it is a inhibitor of GLUT1 and GLUT3, we check the expression of GLUT1 and GLUT3 by RT-PCR, Western blotting, and immunofluorescence staining. The result shows that glutar treatment to tumor cells inhibited the expression of both GLUT1 and GLUT3 in RT-PCR and Western blotting, and also in immunofluorescence staining. Further, the concentration of glucose was higher in culture supernatant. This shows that glutar treatment to tumor cells inhibited the uptake of glucose. Next, we check the expression of key metabolism and cell survival regulatory molecules, in which we found that glutar treatment to tumor cells modulated the expression of various metabolism and cell survival regulatory molecules. Further, we check the expression of pH regulator MCT1 by treating tumor cells with glutar, where we found that glutar treatment to tumor cells inhibited the expression of MCT1 along with increased pH of extracellular pH, and also decrease in the lactate level in culture supernatant. Hence, glutar treatment to tumor cells could also interfere with pH homeostasis. Next, we also check glutar treatment to tumor cells for the ROS expression and mitochondrial membrane potential by DCFDA and TMRA fluorescence staining, where we found that glutar treatment to tumor cells increases ROS expression along with decrease in mitochondrial membrane potential, indicating their involvement in the apoptotic cell death. Glutar was also found to chemosensitize the action of anti-cancer drug cisplatin, both in vitro and in vivo condition. Also, it shows that glutar treatment to tumor cells inhibited the expression of MDR1. The in vitro and in vivo part of the study shows that glutar exposure to tumor cells inhibited the expression of GLUT1, also associated cell survival and metabolism regulatory molecule, and also it alters the pH homeostasis of tumor cells, along with increase in ROS and depolarization of mitochondrial membrane potential leads to induction of apoptotic cell death. It also augments chemosensitivity of tumor cells by altering MDR1 expression. I would like to conclude the oral study. Glutar possesses a promising potential for its antineoplastic action by its catastrophic effect on tumor metabolism, and will also have a long-lasting impact on using it as an anti-cancer therapeutic approach. Some part of the study has been already published and some are in press, and I have also published some research work with my seniors. These are the list of publication. Thank you. Yeah. We have checked the effect of glutar on normal thymocyte cells. No. Yeah. No, it is glutar is like we are using glutar in very less concentration, and as we know that tumor cells overexpress the GLUT1 and GLUT3. So it is only targeted for tumor cells. Yes. Thank you, sir. Yeah, yes. Sir, there will be a reason of some hormonal effect of the gender specific hormone in male and female. We have also done some experiment regarding this, but I have not shown here. I could show if you want. Yes, sir. I have checked the effect of gender hormone on the expression of GLUT1. So I found that gender hormone testosterone and estrogen both augment the expression of GLUT1. So, but the augmentary effect of estrogen, that this female specific hormone, was higher as compared to testosterone. Yes.
glucose is acting as a competitor.
Yes, glucose. Yeah.
So how will you test that?
Sir, actually, I have done in silico study where the active binding site of.
Not bother to the company.
Okay.
In other words.
Otherwise, I have checked another. I have did one experiment in which I have given glucose and fructose to the culture medium of the tumor cells after glucose. Yes, sir.
Biochemical test. You can do studies.
Yes, sir. We can do. We can do. I have not done.
How will you do it?
I have not done, sir.
How will you do it? See whether a compound is an inhibitor, competitor inhibitor, or non-competitor. What will you do?
Sir, actually, we can use the glucose 2DG uptake of the particular 2DG uptake assay,
You can express this protein.
which GLUT1 and GLUT3.
Can we express it heterogeneously, like we active in solution?
No, I don't have any idea about this, sir. Mitochondrial membrane potential. Yes. Sir, I have only checked the mitochondrial membrane potential, but not direct in connection of the mitochondria with the GLUT1.
Any other questions?
Thank you so much, sir.
The next listener is Kumud Tiwari. Kumud is coming from Banaras University. We're talking about the role of transcription factors TTF1 in UVPF damage repair. 8 minutes plus 2 minutes.
Good afternoon, everyone. My name is Kumud Tiwari, working under Dr. Samreen Kumar Singh, School of Biotechnology, Banaras Hindu University. My today's topic is role of transcription factor TTF1 in UV-mediated DNA damage repair. Now, TTF1, that is transcription termination factor 1. It is an essential mammalian transcription factor, and it also known as RNA polymerase 1 termination factor. The gene which encodes is present on the long arm of chromosome 9. TTF1 play a crucial role in different cellular functions like rDNA transcription initiation, termination, chromatin remodeling, DNA blockage. Besides of all these functions, there is always a scope for the novel finding of the novel interactors or the novel functions. So that I will talk in this discussion. So TTF1 become more important when that it play a role in the polar combination of the information termination. So in this figure, you can see this TTF1 outside terminate and by this prevention, it prevents interactions during the rDNA transcription. Similarly, there is a protein which is present in yeast, that is Reb1, which also perform the same function as TTF1 perform here. So TTF1 interact with CSP, Step5, PCAP, helps in chromatin remodeling. Other than that, it interacts with the ARF, MDM2, and help in cell cycle regulation. There are also interactors which interact with the TTF1 and play a specific roles. So for the finding of this novel interaction and function, so for this set of experiment, from the previous knowledge, previous studies and the discovery from Ingrid Room, she is the pioneer of the one. So we found that the truncated this 190 amino acid from TTF1, codon optimize and prepare a construct that is delta 190 and TTF1. So after this construct preparation, we clone this MRQ vector, then subclone into the P0C2X vector and express in BL21DE3. So here you can see the induction profile. After this, we purify this nickel anti column, and lane 5 to 9 shows the elution of this TTF1 protein. So after this purification, it is very important to check that whether the purified protein is homogeneous. So for this, we perform the DLS, that is diffraction like scattering, and in this graph, you can see this protein is a homogeneous state. So with the help of this purified protein, to find the novel interactor, we perform the pull down assay, and with this pull down, we perform the LCMS, and by the LCMS, we got more many different. But the protein DDB1 damaged by protein 1 attracted us because our lab is basically work in DNA damage repair and cell cycle. So with this interaction, we found a novel interactor that is DDB1. So for the check this interaction, we perform the co-immunoprecipitation, and via co-immunoprecipitation, we got that the DDB1 could also cool down TTF1 as well. After this co-immunoprecipitation, now we confirm that DDB1 interacts with TTF. So now let's understand why DDB1 is important, why I'm focusing on this. Let's understand what is DDB1. So DDB1 play a significant role in DNA damage repair, that is nucleotide exchange. So this DDB1 recognize the photolesion sites in global genome, while in TCR, that is transcription coupled NER, the CSF after the sites, TFH binds and excites the damage site, and RNA forms this fills the gap. So other than this, DDB1 also interacts with this ubiquitin ligand system, interacts with DCAP, and by the interaction of these different proteins, it's play a significant role in. So on the basis of that, and I earlier explained you and showed you that TTF1 also finds this interaction with CSP. So we hypothesized that somehow this TTF1 damage repair. So further to check the interaction between DDB1 and TTF1, we further perform this set of experiments. So next set of experiments is, first we silence this TTF1 via siRNA in head to 90 cells and found that after the silencing of TTF1, the DDB1 protein also decreases in head to 93 cells. But interestingly, we found that when we silence the DDB1, the TTF1 interaction decreases in this head to 93 cells. So this results conclude that the DDB1 is the target for the DDB1, but DDB1 is not the target for TTF1. And further, this result should be checked by the ubiquitination process, which we hypothesize is kind of this experiment. So after that, we also want to understand whether it shows any significant effect in UV-mediated immune system. So what we did in this head to 93 cell mimic this 1 minute UV exposure. So first in this figure, you can see the gamma exchange. So it means after 1 minute UV exposure, this system shows the UV mediated DNA damage. So similarly, I performed the same found that after the TTF1 it shows that TTF1 has an important role in UV mediated DNA damage. So this result hypothesized that this TTF1 important during the UV mediated DNA damage repair. After this set of the experiments, I would like to conclude my talk. So first, we stated that DDB1 is a novel interactor of TTF1, and TTF1 is the target of DDB1, and the establishment of role of TTF1 in that is nucleotide excision repair, which is UV mediated DNA damage repair, will open a new aspects for the direction of the DNA damage repair pathway, and also role in different diseases which is caused due to impairment of DNA damage repair. And here I would like to mention that TTF1 is already established in colorectal cancer and lung cancer upregulated. So this we basically focus on the mechanism of this interaction and the specific pathway. So after all this, we successfully can state that significant role in immune-mediated DNA damage repair. I would like to thank my PI, Dr. Samrin Kumar Singh, my lab member, funding sources that is DBT DST. Thank you so much.
Hello, very nice presentation. It's open for discussion.
This is the PDF
Sorry, sir.
written form.
Yes, sir.
Only dictates UV.
Your study, you are mentioning that DCAF that is not interacting with PTF1, and the DCAF3, you are actually cloned the one you are actually mentioned about.
DCAF3. PCNA. Yes, sir.
Yeah.
This DDB1, sir.
Okay, you mentioned about that is not interacting with TTF1. There is another molecule you are actually cloned and identified, no? DDCAF, yeah, DCF. Then that molecule have any interaction with UV or only TTF? That molecule have any interaction with UV or just TTF have interaction? Then what is the rationale you are actually used that DCF for study purpose? How it's not interacting with TTF? No, just want to know that DCAF3 you are actually identified, and that is not interacting with TTAF for like.
You had made a deletion and you had you have made a synthetic construct. How did you make it? Why did you have normal protein was not getting expressed in
we also try not me purify the whole is a big
so this deletion of 190 amino acids.
Yes.
So that has helped in expression.
Yes.
What way? What way has it helped in expression?
Purify the whole Sir, I can also show some. Okay, thank you, sir.
Next presentation is OP26, apoptosis induction in cervical cancer cell by luminoid nilotinib. Anuja Joseph, CSIR, Nash, Ministry of Interdisciplinary Science and Technology, Tiruvandhuram.
Thank you, sir. Good afternoon, all. My name is Anuja Joseph. I'm presenting a part of my research work entitled as Apoptosis Induction in Cervical Cancer Cells by the Luminoid nilotinib. The work has been carried out under the combined guidance of Dr. K.V. Radhakrishnan and Dr. Kaustakumar Maiti, CSIR NIIST, Trivandrum, Kerala. So coming to introduction, nature, as we all know, it is a huge repository of its own molecules for the cure of various diseases. Natural products are the molecules of natural origin. In terms of pharmacotherapy, we can say that these natural products are used in treatment of various diseases like cancer, cardiovascular diseases, etc. And my topic of interest is cancer. Cancer is the uncontrolled growth of abnormal cells anywhere in the body, may arise due to various external or internal factors, and it is the second leading cause of death worldwide. And among that, cervical cancer stood as the second leading cancer in women. And about cancer, like there are various treatment modalities currently available like surgery, chemotherapy, etc. But the problem associated with currently available treatments are that these methods will affect the normal cells also. So we need a particular treatment or a drug which will cause the death of cancer cells but won't affect our normal cells. So to address this problem, the use of natural products in drug development against cancer will be a good solution. And there are already compounds like Taxol, Vinblastin already available in our treatment. So coming to nilotinib, it is a compound not well explored for its anti-cancer property. It is a type of triterpenoid and it is commonly found in the family Meliaceae and Rutaceae. Here we have isolated the compound from Aphanomyces polystachya from its bark. And preliminary cytotoxicity study to check whether the compound is active against cancer cells was done by MTT assay. We have done the assay in HeLa cell line, and the IC50 value after 24 hours of incubation was found out to be 11.65 micromolar. So it is important to check whether this compound is harmful to healthy cells also. So we have done the MTT assay in MCF10A normal cell lines, and the IC50 value was found out to be 83.31 micromolar. So to check whether the compound induces any apoptotic effect on HeLa cells, we have done the ethidium bromide acridine orange dual staining method. Like in this assay, the dead cells in which the cell membrane permeability is lost, this ETBR will cross the membrane and it will stain red. So in increasing concentration, we can see that more red cells, which indicates the dead cells. And for further confirmation, we have done APOP assay. Here also in membrane compromised cells, the stain will be the cells will be stained pink. In increasing concentration, from the result, we can see that more cells are stained pink. So this will route to the confirmation of apoptotic potential of the compound in HeLa cells, which is further confirmed by FITC annexin 5 staining of HeLa cells. In this result, we can see that more apoptotic cells in the increasing concentration comparing to the control cells. And DNA condensation is a major feature of apoptotic cells. So it is confirmed by doing the Hess staining. In this also, the condensed DNA will be fluoresced brightly by this staining method. And by increasing concentration, we can see that more DNA fragments are stained And it is further confirmed by the DNA fragmentation analysis by agrosgel electrophoresis. DNA laddering is a peculiar feature of cancer cells, a peculiar feature of apoptosis on action of nilotinib in HeLa cells. This DNA condensation occurs and endonucleases cleave this DNA into various fragments. And in 12 micromolar concentration, we can see the DNA laddering. And to check about the cell cycle RS, we have done the cell cycle assay the concentration or percentage of cells in the result shows that more cells in the G1 phase, which may be we can confirm it will be the G1 to S phase RS, which should be confirmed further by cell cycle proteins. And to check whether the apoptosis induction in HeLa cells by nilotinib is by intrinsic or extrinsic mode, we have done the fluorometric assays of caspase-3, caspase-9, and caspase-8. And caspase-3 and caspase-9 here shows the it is seen to increase than caspase-8, which confirm the intrinsic apoptotic method, like it is mitochondrial mediated. And as the mitochondrial mediated cell death apoptosis is involved in this one, we have checked the mitochondrial permeability also. Like in apoptotic cells, the mitochondrial permeability or depolymerization is lost. And like in control, we can see that more red cells are seen compared to the treatment. Like more green fluorescence is seen in the treatment, which will show the mitochondrial depolymerization. So as a conclusion in this study, we have done the preliminary assays using MTT and acridine orange ETBR dual staining, and DNA condensation studies were done by Hestine and DNA laddering. Cell cycle assay was performed, and it is confirmed that the intrinsic mode of apoptosis by the activation of caspase-9. And this is the first detailed work of nilotinib on any cancer cell line, and further mode of action is yet to be elucidated. Thank you.
Good presentation, very nice presentation.
Thank you, sir.
And it is open for questions. Questions from audience? Okay. All these studies you are actually conducted with 293T cell line, no?
HeLa cells, sir.
Sorry, HeLa cells. And what about the normal cell lines?
In normal cell lines, only preliminary assay was performed to check whether it is toxic or not. So it was not that much toxic to normal cell line, up to 80 micromolar concentration.
Okay, and not further studies are conducted with like DNA fragmentation?
Not in normal cells, sir.
Okay. And which plant you are actually isolating this particular compound?
From the barks of Aphanomyces polystachya.
Okay. How much the efficiency of this compound you are getting from?
How much?
How you are actually isolate yourself, or you are actually purchasing from somewhere?
No. In our lab, actually, there are both chemistry and biology works are going on already. It is isolated by the seniors.
Okay.
It is not purchased. No. It comes under that class, sir. Nilotinib is the compound.
And previously, any detail, what is the rationale you are selecting this particular compound? Already have any literature available? This compound have any anti-cancer properties are there, or?
From this plant, actually 7 or 8 compounds are isolated, and I have screened that compounds for its anti-cancer potential. And in nilotinib, just one preliminary studies are available in cancer, but no further studies were done.
what is the availability, bioavailability of this particular compound?
Oh. Sorry, sir, I didn't check that.
So you had, no, before this. So you have a 15 micromolar concentration. The best results, or it is only 10 and 12 that is giving best results?
Actually, in 50 micromolar, we'll get good results, but we cannot like perform any assays like cell will be detaching due to heavy death. So at up to 12
So even
micron.
in your DNA fragmentation assays, you have stuck only with 10 and 12 micromolar.
Yeah, I have done in higher concentration, but high, huge, like good cell death is found. So I couldn't perform assays in that.
So these are all on cancer cell line, or these are all on healthy cells?
These are all on cancer cell line.
Only HeLa cells?
Yeah.
You have not tested it in healthy cells?
I have done a preliminary study in healthy cell to check whether it is toxic to
What
healthy cell.
concentration did you take?
Like I have checked from 5 to 100 micromolar and I got 83 as its IC50 value. So up to 83
Micromolar.
yeah,
But
concentration.
here you are saying like 12 micromolar.
No, it is like it is in HeLa cells, like in cancer cells at 12 micromolar, cell death is happening, but not in normal cells. Thank you, sir.
Thank you.
OP 27, Saima Seida from University Delhi. And she is talking about deciphering exosome-mediated crosstalk between tumor cell and macrophages and their polarization in lymphoma.
Respected chairperson, coordinator, and dear participant, good afternoon, everyone. I am Saima Sayada. I am pursuing PhD under supervision of Professor Anju Srivastava from Department of Zoology, University of Delhi. I am presenting part of my thesis entitled Deciphering Exosome-Mediated Crosstalk Between Tumor Cells and Macrophages and Their Polarization in Lymphoma. So to start with exosomes, these are nanovesicles which are normally produced by the normal cells, and they cargo various cargo, nucleic acid, proteins, as well as lipids. So they form a kind of intercellular communication network throughout the body. So in a healthy individual, the number of exosome, it is maintained at basal level. However, its number increases significantly in various pathological conditions, including cancer. So if we talk about cancer, it's a complex process which involves the interaction of tumor cells with homotypic as well as the heterotypic cell types at tumor microenvironment as well as throughout the body. So these tumor cells, they release cargoes, oncogenic cargoes enveloped within these exosomes and mediate the protumoral, alter the target cell behavior towards a protumoral type. So basically, we are working on how these tumor-derived exosomes, they mediate the immune evasion, and particularly how they alter the macrophage polarization or activation. There are various studies which suggest that within tumor microenvironment, there is increased infiltration of macrophages and which is associated with poor prognosis in various cancer types. But how these exosomes, they mediate the polarization of macrophages, it is not much investigated. So if we talk about the macrophages, they are highly plastic cell in the sense that depending upon the environmental stimuli, they can become classically activated to become M1 type, which represents a pro-inflammatory phenotype, and it also shows a cytokilling effect. On the other hand, if the macrophages, they become alternatively activated, they will become M2 type. These M2 macrophages, they promote the tissue remodeling and cell survival. So the report suggests there are contrasting reports. Some few reports suggest there is activation of pro-tumoral M2 type activation of macrophages within tumor microenvironment, but others also suggest there is mixed phenotype of M1 and M2. So we were interested in knowing how these tumor-derived exosomes, they mediate the polarization of macrophages, whether towards M1 type or M2 type. So to investigate this, we use the murine model of Dalton's lymphoma, which is a kind of T-cell lymphoma. We grow the DL cells in the mice peritoneum, and the growth is assessed by body weight and gut size. In the peritoneum, as the DL cell grows, the ascitic fluid accumulates within the peritoneum. So from that ascitic fluid, which represents the rich source of tumor factors, we isolated the exosomes, and then further we check the effect of these exosomes on macrophage polarization. So firstly, we evaluated the murine model for the presence of exosome. So firstly, we performed the staining of the tumor cells with exosome specific marker, wherein we observed the high increased biogenesis of exosomes. Further, we check the exosome burden in the tumor microenvironment, which is the peritoneum, and we observe that within in the peritoneum section of the DL or the tumor bearing host, it is the level of exosome increased. So after evaluating the tumoral exosome burden in tumor model, we isolated the exosome from the ascitic fluid and characterized them using various techniques. Firstly, by electron microscopy, which represent the vesicle-like structure in our sample preparation. Further, we check the size using the dynamic light scattering. The exosome size varies from 30 to 150 nanometer. In this our sample is also the exosome in our sample also lies within that range only. Then we further characterize the exosome using exosome specific markers, and we found that in our preparation of exosome, the exosome specific marker shows the highest expression. So after characterizing the exosomes, we performed the in vitro study using the murine macrophage cell line, which is raw cells. So before looking into the effect, we check whether these exosomes, the isolated exosome, they are taken up by the macrophages or not. So we performed the exosome uptake assay, in which we stain the exosomes before incubating with macrophages and at different time points. So we observe that with increasing time points, the exosome uptake has been increased by the macrophages. So after evaluating the uptake of exosome by macrophages, we set our experiment and we took 4 groups: control group, second exosome incubated group, and third is the LPS stimulated group. LPS represents the inflammatory condition which activates the macrophages towards M1 type. So in tumor condition, we hypothesize that the exosome might be activating the macrophages towards M2 type. So we included LPS to differentiate between the M1 and M2 characteristic. Then we studied various parameters, including morphology, wherein we observed a contrasting difference between exosome incubated macrophages versus the control group. Also in the LPS stimulated group versus the exosome group, we observe a contrasting difference, wherein in exosome incubated group, we observe the morphology of the macrophages become elongated type. On the other hand, the LPS stimulated group, in that group, the macrophages show more flattened phenotype. Next, we look for the reactive oxygen species level. During inflammatory condition, it is very much high, but in cancer condition also, the reactive oxygen species level, it promotes the tumor growth and survival. So here also, we observe the increased level of reactive oxygen species in macrophages incubated with exosome. Next, we looked for the another important parameter, which is the L-arginine metabolism. So L-arginine is a common substrate for the two competitive enzyme, nitric oxide synthase 2, NOS2, and the arginase 1. M1 macrophages, they express the NOS2. On the other hand, the M2 macrophages, they express the arginase 1. So for this, the NOS2, it metabolites L-arginine into nitric oxide. On the other hand, the arginase 1, it metabolites L-arginine into urea or in the polyamine synthesis, which is important for tumor cell survival. So when we estimated the nitrite level, we observed that in the LPS stimulated group, which represents the inflammatory condition, it is showing the high level of nitrite release. On the other hand, when the LPS plus exosomes were treated with the macrophages, then the exosome mediated or reduced the LPS induced NO level in the macrophages. Further, we looked for the enzyme level, whether the NOS2 and arginase 1, they are expressed by the exosome incubated macrophages or not. So when we looked for the NOS2 enzyme level, it was found that in the LPS stimulated group, NOS2 level was significantly high, which reduced upon the with the exosome condition. And further, the arginase 1, its competitive enzyme, it was expressed by the macrophages. Then we looked for the cytokine profile, which is an important parameter to differentiate between M1 and M2. So we observed pro-inflammatory cytokines, IL-12 and TNF-alpha, which was low. On the other hand, IL-6 and IL-1 beta, which is also expressed by M2 macrophages also, so it was high. On the other hand, the anti-inflammatory cytokine, which are specific for M2 type, so they are high in the exosome incubated group. So looking at the cytokine profile, we looked for the TLR profiling because the report suggests that this IL-6 and TNF-alpha pro-inflammatory cytokines, they are released by certain subsets of M2 macrophages upon TLR activation. So looking on the TLR profiling, we observe the high expression of TLR2 in our study. So from here, we can conclude that the lymphoma-derived exosome, they activate the macrophages towards pro-tumoral M2 type by activating the TLR2 activation. So further, this could be targeted by using potent immunomodulatory agent or nutraceuticals for immunotherapy in cancer management. Thank you, sir.
Thank you, Saima.
Yes.
Very nice presentation.
Thank you, sir.
And it's open for questions.
Regarding exosome. Yes. Yes, sir. Sir, in liver, in what aspect? Sir, we observe the splenomegaly, like enlargement of spleen, not the liver. But the up to 4 hours. For DL, for staining the tumor cells or for the uptake assay. For uptake assay, we stain the exosomes using the PKH stain using a kit. No, sir. It's a dye, PKH dye, which stains the plasma membrane.
Which method you are actually used for isolation?
So
exosome
using
isolation?
commercially available kit from
Okay.
in vitro.
And could you
exosome
check the
isolation kit.
efficiency, like compared to the centrifugation method, this is the best method? Or what do you think about it?
Sir, centrifugation method nowadays, the comparison has been made, and the exosome isolation kit are referred to be the best over the ultra centrifugation methods. And in the characterization also, we observe a good number of exosome in our preparation. So that is why we did not look for the ultra centrifugation method.
And you mentioned about there will be exosome, exosome with macrophages, there will be elongation is actually happening. And exosome morphology is actually changing.
Yes, yes.
And what will be the reason for that? How this is actually possible?
Sir, actually it is getting activated. So upon activation, it changes. There might be like the reason. Sir, upon activation, the macrophages has the ability to change its phenotype. Like upon LPS stimulation also, it changes its morphology towards more flattened type to become more phagocytic in nature. On the other hand, the M2 macrophages, they do not show, they show the reduced phagocytic activity compared to the LPS stimulated macrophages.
Thank you.
Thank you, sir.
Molecular toxin study suggests that menstrual disorder caused with PCOS may be treated with Himalayan medicinal plant Xanthosylum.
Very good afternoon to one and all. My name is Vinita Sharma. Currently, I am pursuing my PhD under the guidance of Dr. Shankar Mandal from LSM Government PG College, Pithoragarh, Uttarakhand. First of all, I would like to convey my sincerely gratitude to the organizing committee for providing me this opportunity to present my work in front of respected chairperson and my senior research colleagues. So today I'm going to talking about a molecular docking study suggests that menstrual disorder caused by polycystic ovarian syndrome may be treated with Himalayan medicinal plant Xanthosylum aromatum, which is commonly known as Timur. This presentation will focus on 4 points, that is some basic introduction, then materials and method, result, and finally discussion. So as the picture shows the birth and developmental stages of women, and we all know that all human beings shows 2 vital signs, that is metabolism and reproduction. In reproductive ages of women, basically this reproductive period of women is a period between the menarche and the menopause, in which menarche we know that there is an onset of menstruation in which one ovum is released from the ovary, and if not fertilized, it passes through the reproductive tract during menstrual cycle. So what happens during menstrual cycle? Here we can see in this picture that the 2 portions, blue one and the pink one, in the upper side of portion, that is ovarian activity, in which menstrual cycle is categorized into the 3 phases. That is the menstrual phase, which is characterized by the maturation of follicles, and then the ovulatory phase, the phase of ovulation, and then the luteal phase, in which the mature follicle transforms into the Graafian follicle, and this phase marked by the end of uterine bleeding, and then the menstruation occurs. The lower portion shows the uterine activity, in which the first one is proliferative phase, in which endometrium rapidly increases under the influence of estrogen. Then the ovulation occurs, and after that the endometrium increases its secretory activity under the influence of progesterone as well as estrogen. Now, in endocrine point of view, if we see, in response to gonadotropin releasing hormone, anterior pituitary secretes 2 types of gonadotropins, that is follicle stimulating hormone and luteinizing hormone. Follicle stimulating hormone acts on ovary, which stimulates the development of follicles, and these follicles surrounded by 2 types of cells, that is the theca cells and the granulosa cells, which we will discuss in the next slides. After the maturation of follicle development, the final follicle is a Graafian follicle, which generates estrogen level and which ultimately signals the pituitary gland to seize that FSH so that it can stop the further maturation of follicles. But on the other hand, it gives signals to pituitary to release luteinizing hormone for more and more release of luteinizing hormone to occur ovulation, and the process continues. But the reason behind showing this pathway is that the whole process is the interplay of hormones in which estrogen and progesterone are formed, which is an steroid, and these secretion is formed due to a process called steroidogenesis. Steroidogenesis, if we talk about in a normal female, is a 2-cell, 2 gonadotropin hypothesis, in which 2 cell means the theca cell and the granulosa cell, and gonadotropin means that the luteinizing hormone and the FSH. So here we can see the theca cell and the granulosa cell. So what happens is that inside the theca cell, with the help of the precursor cholesterol, it forms a pregnenolone with the help of these enzymes, and then into the DHEA and progesterone, and finally it forms androgens. The 2 common androgens here it is shown that androstenedione and testosterone with the help of the enzyme CYP17A1 and HSD. Now, with the help of the basement membrane, these androgens then travel to the granulosa cell, and under the influence of follicle stimulating hormone, these androgens transforms into the estrogen. But in women of polycystic ovarian syndrome, the coordination of hypothalamus, pituitary, and ovary axis does not active properly, which affect the gonadotropin release and which increase the activity of luteinizing hormone, which ultimately stimulates the theca cell to produce more and more number of androgen. Excess amount of androgen may causes the follicles may hindrance in the normal follicular growth, ovulation, and maturation, and then the ovary become compromised with many small antral follicles which never become dominant, and it becomes a battery of symptoms like hirsutism, acne, alopecia, and irregular menstrual cycles, infertility, miscarriages, and various obstetrical complications. Here we can see the normal ovary and the ovary which is arrested antral follicles of polycystic ovarian syndrome. Now coming to this slide, here showing the hyperandrogenemia, how these cells, how this hyperandrogenemia affected by the oversecretion of steroidogenesis pathway. Here the three receptors being used as a target to the first one is CYP1720 lyase, 5-alpha reductase, and human androgen receptor with a drug that is used like metformin and spironolactone and cyproterine reductase. But these drugs that are currently used to treat PCOD shows many side effects like vomiting, fatigue, irregular menstrual cycle. So a new drug molecule, a natural ligand should be searched for the treatment which have a potential effect without side effect. So the aim, according to the factors and to look this consideration, the aim of a study is to virtually screen the phytochemicals present in Xanthoxylum armatum, which may potentially inhibit the steroidogenesis pathway. So the main focus of our study was to inhibit these receptors with the help of natural ligands of Xanthoxylum armatum and to compare it with the reference drug like metformin, spironolactone, and cypriterone acetate. For this, the objectives of our study is to search the phytochemicals present in the Himalayan medicinal plant Xanthoxylum and to perform in silico screen of these phytochemicals and to compare the binding energy of phytochemicals with the standard drug. To fulfill the objectives of this study, first of all, the three-dimensional 3D structure of these receptors were retrieved from the protein data bank, PDB. PDB is the largest protein bank from where we can download these structures and we can retrieve the PDB ID. Then the ligand preparation, the structure of reference drug and the phytochemical compound of Xanthoxylum armatum were retrieved from the PubChem databases. After the retrieval of these ligands and the receptor, molecular docking was performed by the help of software that is Pyrex. So based on the highest binding affinity, we have found the 18 phytochemicals compounds of Xanthoxylum as the table shows, in which the table shows the binding score, docking score of these phytochemicals, in which we can see if here the first one is hesperidine showing minus 10.1 kilocalorie with CYP17 and minus 12 kilocalorie with 5-alpha reductase receptor. Similarly, if we see here with lupiol, it is minus 10.2 and 11.2 with 5-alpha reductase, not 1 and 2, but all the 18 phytochemicals showing highest docking score. More negative the docking score is, it shows, it represents the more strong affinity. So if we compare it with the current drug that are currently using for the treatment of PCOD, that is metformin, it shows only minus 5.1 kilocalorie with CYP17 and minus 5.1 for 5-alpha reductase. And if we see here is spironolactone, it is minus 8.6, and for cyproterone acetate, it is minus 9.6. So if we conclude that here the two pathway where we can say that the phytochemicals that we have used with Xanthoxylum aromatum is effectively used to treat the PCOD without any side effects. And this is the summary of our result, in which 58 publicly available structures of phytochemicals of Xanthoxylum aromatum screen. 18 compounds shows highest binding efficacy. So at last, I would like to conclude by saying that we had taken FDA approved drugs that I have already told, that metformin, but it shows the side effects, that is infertility and many more. And we have used that natural ligand phytochemical, which is effectively work in this PCOS. And also we have used the in silico screen, which is very time consuming and a good result. Thank you.
Yes. Yes.
Actually, the ligand that I'm talking about, hesperidine, is a natural ligand that extracted from a medicinal plant, that is Timur, which have no side effect. If we see the toxicity of the Timur, it shows that there is no side effects against that PCOD in comparison to if we talk about the metformin or spironolactone and cyproterone acetate. That is why I'm talking about that. We have shown the result, which is effectively work in comparison to the synthetic drug that are currently used for PCOS.
What is the rationale you have selected?
Actually, I'm working in Uttarakhand, which is very widely, which is a place where these medicinal plants are widely used and easily available plant that people used as a natural home remedy for the people that are living in interior places. They always use this kind of medicinal plant for the treatment of their menstruation, menstrual cramp. So that I think about that. And it is very widely used for the stomach ache and menstrual cramps.
Thank you. From Nirmala D.S. Gowda, and she is going to be talking on quercetin, and quercetin ameliorates metabolic syndrome associated with non-alcoholic steatohepatitis via adiponectin.
Yes, sir. Thank you, sir. Good afternoon, all. So I am Nirmala GS from Jesus Medical College, Mysuru. I am currently pursuing my PhD under the guidance of Dr. Prasanna K. Santakaduru. So today I am going to present my research topic on quercetin ameliorates metabolic syndrome associated non-alcoholic steatohepatitis via adiponectin. So before going to result part, let's have an introduction. So very first, what is metabolic syndrome? Metabolic syndrome is a cluster of interconnected diseases that include the risk of cardiovascular disease associated with obesity, type 2 diabetes, dyslipidemia, and NAFL, non-alcoholic fatty liver. So, like, why this metabolic syndrome is increasing day by day? So there are some factors like sedentary lifestyle and physical inactivity and overweight. So these are some factors why the metabolic syndrome is increasing day by day. And these are the list of
things which involved in the metabolic syndrome, like nutrient deficiency, metabolic defects, diet and lifestyle factors, environment and genetics. So in my work, I'm focusing on the diet and lifestyle factors. So can we recover a metabolic syndrome? Yes, by adopting some healthy habits, we correct our lifestyle pattern, balance our calorie intake, and control our weight gain. So these are the things which can be adopted to recover the metabolic syndrome. So there are very few drugs which are available in the market for the non-alcoholic steatohepatitis associated metabolic syndrome, and these drugs have a side effect. So to limit the usage of these drugs, so the natural compounds is the best to adopt further as a therapeutic target. So discovery of natural agents with antioxidant and anti-inflammatory effect without any side effects to treat metabolic related diseases is one of the challenging role. So in my work, I choose quercetin. Quercetin is a plant flavonoid which abundantly present in various fruits and vegetables, and which has a various medicinal properties too. So if our diet, like eat your food like a medicine. So we know the diet pattern and our lifestyle. So we can change our disease, like we can change our lifestyle to control or prevent the disease condition. So coming to the results and discussion part, very first, we in vivo study, we take the Swiss albino mice, which is 5 weeks old, and up to 8 weeks, we given the one group will given with chaw diet, and that is what standard diet, and another group fed with a high-fat diet, and the next group from the 8 weeks to 12 weeks group administrated with a quercetin up to 12 weeks, and then the another group will administrated of quercetin up to 16 weeks. So when after the sacrifice, so we observe the basic observation. So that is what in the case of high-fat diet fed mice, there is increase in the body weight compared to chaw diet. And in the quercetin treated group to 12 weeks and 16 weeks, there is a reduction in the body weight compared to high-fat diet. So the graph depicts the body weight. And also we observe the organs weight also, that is what in the case of obesity, that is what adipose tissue as well as liver. So we can see the in liver the fat accumulation we can observe in high fat diet compared to chaw diet, and in the treated with quercetin to 12 weeks 16 weeks we observe the less fat accumulation and the changes of architecture of liver also, and the graph depicts the liver weight. So next we did perform the in vitro study in 3T3L1 cell lines. Even though I'm since talking about the metabolic syndrome, I'm here talking about the crosstalk mechanism between the heart, liver and adipose tissue. So to mimic the obesity model, I choose the 3T3L1 pre-adipocyte. So by giving the components, I differentiated the pre-adipocyte to the mature adipocyte. So, and I performed the oil red dose staining to know whether my quercetin compound will inhibit the adipogenesis in 3T3L1. And also I performed, check the glycerol released to whether to know the quercetin compound inhibits the enhance the adipolysis. And also I measure the intracellular TAC content also in 3T3L1. So this study, this data reveals as quercetin will inhibit the effect of fat accumulation in the mature adipocyte, and also it enhances the adipolysis lysis, and also it decreases the intracellular TAC content also, that is what triglyceride. So coming to the quercetin amyloids lipid profile induced by high-fat diet and high-fat diet fed mice. So even though you are talking about the obesity, like triglyceride, cholesterol, and LDL, so these are the parameters of the lipid profile. So when I measured these parameters of lipid profile, I observed the significant increase in the high-fat diet content, high-fat fed mice compared to chaw diet. And in the case of quercetin treated, there is a reduction in the these parameters. And very interestingly, HDL is also decreased in high-fat diet, and when quercetin treated group, the HDL level is increased. So HDL is a good cholesterol. And then I performed the qPCR assay study to elevate the level of mRNA level of various inflammatory markers in adipose tissue like IL-1 beta, TNF alpha, IL-6 and MCP1. So in that also quercetin treated group, there is a a significant decrease in the mRNA expression of these inflammatory markers. So when adipose tissue, what happened when the inflammatory markers will increases, that leads to cause a various disease condition too. So in order to from this study, we next take out the effect of quercetin in cardiac inflammation and fibrosis. So first I did HND staining and MTS staining. HND staining is for the inflammatory to check the infiltration level, and MTS for the fibrosis level. So when we did for the HND and MTS in high-fed mice group, shows a more immune cells infiltration in HND, and in MTS we can observe the blue color. So that is a deposition of collagen. So when treated mice quercetin treated group, there is a reduction in the inflammatory infiltration cells and also in the fibrotic levels. So next I did the QRT-PCR in heart tissue. I did the inflammatory markers IL-1 beta and TNF alpha. So in that also quercetin gradually reduces the inflammatory markers in cardiac tissue, as well as I checked the collagen markers also, and these are the targeted genes of TGF beta also, collagen type 1, type 2 and alpha SMA. So in that also quercetin significantly reduces the fibrotic markers in heart tissue. So next I connected to the liver, because when the excess fat accumulated in the adipose tissue, so these excess fat will circulate and it accumulated in the liver, and which causes a non-alcoholic fatty liver. Then it causes inflammation and it causes fibrosis and leads to causes various disease stages of liver disease. So first I performed the in vitro study. So by giving sodium palmitate and oleic acid as a fatty acid treatment in HAS7 and FG2 hepatic cell lines. So here also I observed that in quercetin treated group there is a less accumulation of lipid droplets. So next I performed the HND staining and MTS. In that also we can see the hyperdiet hepatic cell ballooning we can observe, and also the fat accumulation. This blue color indicates the deposition of collagen. So in the quercetin treated group, there is a reduction in the ballooning of hepatocyte as well as fibrosis. So next, I've measured the liver enzymes, because inflamed liver will elevate the level of liver enzymes. So in that also, the quercetin treated group decreases the level of different liver enzymes too. And also I measured the hepatic triglyceride, also cholesterol. In that also quercetin reduces the hepatic triglyceride as well as cholesterol. So when we come to how the quercetin will regulate the cardiac inflammation or hepatic inflammation. So then we got adiponectin. So adiponectin is a fat derived hormone. So when in quercetin treated group, that is in the cell line study, and this is in the serum adiponectin of mice. So in that also quercetin treated group, adiponectin level is enhanced. So then we measure the adiponectin receptor 2. Is the adiponectin receptor 2 is the receptor for the adiponectin. We measured in the hepatic liver tissue as well as in the heart tissue. In that also quercetin increases the expression of hepatic adiponectin receptor 2. And finally, I measured the fibrosis markers, also TGF beta markers in liver tissue. In that also, fibrosis level will decreased in the quercetin treated group. So by this results, I concluded my talk. When taking high-fat diet, so the adiponectin level will be less. So that leads to cause the lipogenesis, triglyceride content inflammation, and the adiponectin receptor 2 expression is also very low. So that leads to cause the cardiac inflammation and in liver inflammation also. So this study revealed a novel link between the pathophysiological interaction, and given with the quercetin, it enhances the adiponectin level and expression of adiponectin receptor 2 also. So quercetin can be used as a promising drug to treat the metabolic syndrome, and also adiponectin can be a novel and potent biomarker to treat the metabolic syndrome associated NASH. So I would like to thank my guide and my lab mate and all the funding agencies. Thank you.
Thank you. Any questions? Yeah.
That is what 5, 10, 20. 5 micromolar, 10 micromolar, and 20 micromolar for in vitro study. High-fat. Like not the effect of diet, like up to 8 weeks we given the high fat diet. So after also we continued the high fat diet. We are not stopped the high fat diet after up to 16 weeks also, and the 12 weeks, the interval of time interval. So we given the continue with the high fat diet, but after 8 weeks we added the quercetin. Up to 12 weeks one group, and up to 12 weeks 16 weeks. So along with the high fat diet only, how our quercetin act upon the, whether it is helps in the reduction of inflammation or fibrosis, or it will be involved in the like inhibiting the accumulation of fat, or that is what we observed.
Thank you.
Okay, thank you, sir.
Good afternoon, everyone. I'm so sorry.
Next presentation is Neha Mary from Women's Christian College, Chennai. And she is talking about a detailed study on microbial composition and health benefit of fermented food of different zones of India.
Thank you, sir. Good afternoon, everyone. I'm Neha Mary. I'm a PhD research scholar in the Department of Biotechnology, Women's Christian College, Chennai. So today I'll be presenting a part of my PhD project titled Detailed Study on the Microbial Composition and Health Benefits of Fermented Food of Different Zones of India. This focuses on understanding the microbial composition of various non-commercial fermented food of different zones of India and understanding its probiotic effects on human gut and overall health. The sample that I'll be talking about today is called Henta. It's a fermented food from the state of Manipur, made of Indian flying barb fish and petioles of aroid plants. The isolation of the microorganism was done following standard protocol using MRS-agar, which promotes the growth of microorganisms associated with probiotic characteristics. Its antibacterial substance production was checked against a set of gram-positive and gram-negative bacteria that are associated with causing various infections in human, mostly food and waterborne pathogens. Gram-positive being Staphylococcus epidermis, Micrococcus luteus, gram-negative being Yersinia enterocolitica, Shigella flexneri, and Proteus vulgaris. The microorganism I isolated showed good results against all the 5. The identification of the bacteria was performed using different sets of experiments. It was found to be gram-positive in nature, non-spore forming. A series of different biochemical tests were performed to understand the genus of the microorganism isolated, its potential to ferment different saccharides, sucrose, mannitol, glucose, arabinose, and trehalose was also done. And it was found, and it showed positive results to ferment all the saccharides. 16S rRNA sequencing was done to understand and find out the species of the organism. And it was found to be Lactobacillus brevis strain LAP2. The multiple antibiotic resistance MAR index was checked against 7 standard antibiotics that are used to treat various human infections: streptomycin, kanamycin, nalidixic acid, rifampicin, chloramphenicol, gentamicin, and penicillin. And it showed resistance against nalidixic acid, and the MAR index value was calculated as 0.142. The characterization of cell-free neutralized supernatant was done. The organism was grown in MRS broth culture, centrifuged at 2000 RPM, and the supernatant was collected, and its stability was checked. pH stability was checked at both acidic and alkaline medium, and it was found to be very stable against the similar set of indicator bacteria. The thermal stability was checked at 60, 100, and 121 degrees Celsius at 15 minutes, 30 minutes, and 60 minutes, the control being the unheated sample, and it was found to be unstable at heated temperatures. Proteolytic enzyme activity was checked when the sample was treated with pepsin, and the pepsin in the media was able to degrade the protein present in the microorganisms. A set of different experiments were performed to confirm its probiotic characteristics. These set of experiments were basically done to mimic the environment of our own gut flora. Cell viability at low pH, because our own gut flora is acidic in nature, and the cells were viable at 3, 4, and 5, showing that it was able to survive the acidic pH. Resistance to simulated gastric juice, and to mimic the gastric juice, I used pepsin, NaCl, and dipotassium phosphate, and it showed resistance. Tolerance to bile was checked, and the bile salt used was sodium taurocolate, and the readings were taken at 600 nanometer. Proteolytic activity was checked using skim milk agar, which was given peptone and beef extract, and it showed that it was able to degrade the protease present in the skim milk agar. Its autoaggregation was checked, which shows that it will inhibit the growth of biofilm produced by human pathogens after infection. Hydrophobicity was checked, which confirms its binding efficacy to the epithelial cells of our intestines. Antioxidative properties were again checked against standard free radicals DTPH at 517 nanometers. And again with the hydrogen peroxide, which is also a free radical. And the standard used here is vitamin C. So the conclusion is that the study shows that the isolated bacteria Lactobacillus brevis can withstand the environment of the human gut, leading to its finding as a potential probiotic. And the antibiotic susceptibility pattern shows that it's safe to be consumed and can be used in the food industry. And it's a potential candidate for reducing the oxidative stress associated diseases in humans, which claims that it has anti-cancer properties as well. Thank you. Lactobacillus brevis, sir. So 16 is rRNA sequencing. So this is actually just a preliminary study. This is just a minor part of my PhD project. It's ongoing. So I am doing a comparative study, basically focusing on non-commercial fermented food. So I'm aiming to form a bioconjugation of proteins isolated from these microorganisms, because there are studies that shows that even when when probiotics are given to patients with immunocompromised conditions, they sometimes induce lactobacillus induced bacteremia. So no, this is not, sir. This is just done. Yeah, this is the preliminary study to make sure that this strain of bacteria is safe to consume and can be given to people. So the further studies are still ongoing. Yes, sir. I'm sure it is there, sir. This lactobacillus is, I have done work with curd, milk, and few more fermented food, some commercial, some non-commercial, that I haven't mentioned in the work because this one is complete. This is safe. So I presented this work. Yes, sir. Yes. It is only one, nalidixic acid. So that shows that the pattern shows that it can be used in the food industry as well. It has resistance only to nalidixic acid, sir. No. It is, the S means susceptible. Yes. No. It's still sensitive towards others. Yes, sir. Thank you so much.